by which viruses go into these host cells, kill them, release new progeny, and so on. Today I'm going to shift gears a little bit and go from lysis to latency and begin to explore ways in which viruses need not kill their host cells in order to reproduce and the consequences that may have in the long term for evolution. And I would say that this talk is the culmination of thinking for a long time about a problem, and I'll maybe give a little bit of the backstory, and feeling absolutely unable to make any headway until a certain point when I started to think a bit differently. And I might talk about that too. And now, frankly, after Paul's talk about these egalitarian transitions, I need to think a little bit more about whether that's happening in our systems as well. So the thanks slide I want to put very early because this is really something that I've been thinking about, as I said, for quite a long time, which I'll mention even from the very start of my shift from working in a physics PhD and then starting in ecology, evolutionary biology postdoc. And it was one of the first problems I thought about is this question of lysis and latency. And the transition has happened really for me in my thinking with the result of interactions with many folks. And I want to thank people in my group who I'll mention at various times. And there are a few in particular who have done some of the calculations related to today's talk. Wan Lin Lee, a PhD student in my group, Ria Gulbadak, a former postdoc, and Michael Cortez, also a former postdoc, both of whom have their own positions now, as well as collaborators, and particularly Rachel Whitaker at University of Illinois Urbana-Champaign. And also I want to give a lot of thanks to interactions at a workshop in Portugal called a Continuum Persistence Workshop. There are also philosophers there when we were talking about the ways in which viruses persist along a continuum. And that certainly was very influential in the way that I have been thinking and am trying to push ahead. Okay, so let me go back again to remind you this is this life of a bacterial virus that I described. You have this passive diffusion, contact, injection of genetic material into the cell, replication, take over the cell machinery, and then release of new progeny, leading to the death of the cell. So here there's an obligate death of the cell in order to ensure the production of new viral particles, these new virions that can go on to find new cells. And at the very beginning of my first talk, I introduced the idea that because of this way in which a virus kills the cell to make more of it, there was an analogy to be made between predators and prey, which went back all the way to 1961 and 1977, this work of Campbell and Bruce Levin, the idea that there's a simple predator, the virus, and a simple prey, the host. Okay. And so therefore, if one thinks at the very start when there's a susceptible population that a new virus arrives and there's many cells, and we count viruses in terms of these virus particles, then we start with one virus, it kills the cell, and out come hundreds of new progeny viruses. And in that sense, at the individual scale, this burst size seems to suggest that the predator fitness is quite large, right? It made 100 new progeny. So it's amplified its numbers by this very large amount. And of course, there's also this notion of predator fitness at the population scale. And I was in those lectures telling you the difference between them, right? The growth rate of the population, whereas we have a criteria for when that's positive, which has to do with the average number of these new viral progeny per initial. And so we have this individual notion of invasion fitness and a population or notion of invasion fitness, which has to do with the net growth rate of the virus particles. Okay, so we have viruses that kill cells, and we can see that they can spread within populations. But the question I want to ask today is, do viruses and microbes do more than kill or prepare to kill? Right? So everything is at all just oriented around this antagonistic interactions culminating in death, or are there some alternatives? Okay. So there are alternatives. And even for viruses of microbes, there is a classic example, phage lambda is perhaps the most well-studied, though there are others, of bacteriophage that inject the genetic material into the microbial cell. And then rather than directing that cell to make more virus, which then self-assemble and so on, leading to the death of that host cell and the release of new viral progeny, there's an integration event which you can see here, which is called lysogeny, in which the viral genome, here denoted in red, integrates into the host chromosome. And then when the host divides, it carries with it the viral genome, and in that state it's called a prophage for these bacteriophage. And this can happen multiple times. And there can be various reasons why later an event called induction happens. And I'll describe just a few slides some of the conditions where that might happen. And then the virus excises itself from the genome and reinitiates the lytic pathway, leading to the production of more viruses, the death of the host cell, and then the life cycle continues. Okay? So you can already see here that we have an interesting dilemma. And there are really two dilemmas. When the virus of this kind, these are called temperate phage, which have an option to kill or to integrate and form a lysogen, as opposed to virulent phage, which are, especially obligate virulent phage, can only kill their host cell. There's, in some sense, a decision or a switch here. Does one integrate or kill? And then once the virus or prophage is now inside, does one continue to stick with the host, or does one go out, kill the host cell, and try to find a new host? So the way that these things have been looked at, and this has been studied for decades, literally, and modern tools have helped to take this question and bring it all the way down to a single cell perspective. So I'm just going to give a little bit of that background here. This is an engineered strain of bacteriophage lambda, which expresses this M. cherry in its genome insofar as it has integrated into the host cell. So I'm not going to spend this entire lecture on the sort of genetic switch of phage lambda, because that will get us a little off track. But it has a switch, and it has a decision-making machinery, and when it integrates, it turns on a promoter, and they've just genetically engineered the phage so that when it integrates, it's going to also express this M. cherry. And we'll notice then that cells that have been lysogenized will turn red. However, you can also YFP label these phage such that they express stably with the capsid, so we have infecting phage, and you see lytic cells producing new phage. So first of all, you can track individual events, and you can find out how many and where, right, and how big the cell was when an individual phage managed to absorb. And then you can see the time, this is about 80 minutes later. In those cases where a lytic event is happening, you see the production of new phage, and here you see cell lysis. So the cell disappears, and you have a lot of green, new phage, and on these other, you see that there's an integration event, and now the cell can divide, and you can see then clusters of red cells as you have these lysogens vertically transmitting the phage genome from one generation to the next. Okay? And this is work, Ido Golding reviewed it, but he's done quite a lot of this single cell work. He's one of the innovators. He's at Baylor College of Medicine in Texas. And there are many other people working on this. And there's quite a lot of interesting things you can ask then. First of all, there's some genetic circuit here that underlies this decision. And so one of the interesting things that people have done is modify the intracellular viral concentration or the multiplicity of infection and ask, when there are more viruses per cell, this is going to the right here, is it likely for the outcome to be more likely to be lysogenic or lytic, right? And so if I were to ask any of you, do you want to have someone who has the flu or the common cold sneeze on you and get more viral load or less viral load? You, I think, would tell me, please give me less viral load, right? Because you would expect the outcome would be better. Here, this percent lysogeny, the more viruses per cell for this phage lambda, the more likely it is that those viruses will not kill that cell. And this seems a bit paradoxical, and that in some sense brings me back to why I'm even giving this talk, was when I first started thinking about phage-bacteria interactions, and I had wondered, how is this even possible? Right? So if these were independent decisions, it would seem that killing trumps not killing. If I decide to kill, and Marcus gets it, you know, you're a different phage, we both get in, you're going to hang out with the cell, well, too late, it's gone. Now, if we're both the same kind of virus, the way that these decisions are actually made is that they express regulatory proteins And if we're the same, then your regulatory protein may actually influence my decision and vice versa. So it turns out there's a very interesting intracellular decision process, which could be a form of collective decision-making insofar as that they share the same genetic circuits. So that's one interesting puzzle that we'll get back to. I can't claim to have solved it. These are really how questions, how these things happen. And the, yeah.
Is there any significance of lots killing?
Yeah, so I'm going to go in the next slide talk about it. And these are really tantalizing because we're beginning, this is a question that I wanted to answer back in maybe 2003. What do we have? It's 15 years plus. And there are reasons why I always failed at doing it, and I think, and I'll show you in a moment, I think collectively we haven't quite figured it out. Because of the way that we think about fitness and what is adaptive or beneficial to viruses. And so this is some of the things that we're working on now. But I want to point it out here because it's been one of the, going even back to the 70s in Philip Kurilski's work. So this was an old observation. I killed some of those, I took some of the slides away. But there's an old observation of exactly this, going all the way back to the 70s. This has been known for 40 plus years. And so let me tell you in the next slide what I think that what the canonical idea of the adaptive benefit is, but it has a bit of group selection to it, and not in the sense that you've described. So let me just come back to it one more slide. So the other thing and feature that is likely relevant here is that, so this is this initial integration switch. Somehow the first switch somehow depends on how many viruses there are. But once it's integrated, it turns out that if you expose these cells to UV and you begin to, in some sense, damage the host, the fraction of these hosts that are induced, meaning they have viruses in them, then they will start to come out of the host cell and release new progeny. And this, certainly the mechanism there seems to be adaptive, and that one's a clear story. There's an SOS response, which is meant to repair DNA damage. It turns out that that SOS response also binds one of the regulatory proteins and therefore tilts the switch from being stably integrated to one that's lytic. And then, in some sense, the virus is listening in on the state of the damage to the cell. And in some sense, abandoning a sinking ship, as it were. Right? So that one seems clear. This other one, a little bit less so. So these are lessons from a simple system phage lambda over 70 years. I mean, a long period of time. And in the 1980s, I'll get now to the question, I think, Paul, that you're raising. Frank Stewart and Bruce Levin had begun to ask in a much more formal way, a rigorous way, well, why be temperate? Why go through and stick with your host? And I'm going to try to raise this point, which is that I've told you, and it was even asked in, there was a little session here of trying to talk to the speakers and ask some basic questions. It was already asked, what is a virus? And having written a book on a virus and looking up definitions, in my mind, I thought of the charismatic version of virus, which is the particle, and it has neat shapes and so on. And so therefore, if that's the virus, then viral fitness is making that mature organism, which is this particle, right? And so therefore, if you're a virus, you get into a host cell, you could make 100 more viruses right now, or stick with the cell, integrate your genome, but that's still not making virus yet, and hope that one day you come out and make more virus. Now, it's true that they're spreading through a population in some way, the genome is, but I found that a little bit problematic. Why would I not just make a lot of more virus right now? And could not necessarily construct models of invasion that seem to account for the benefit of integration. And maybe you start to say, well, the cell could potentially see a way up, but it always seemed arbitrary. So Frank Stewart and Bruce Levin began to ask this question and also used similar models to the ones I described. They had resources, lysogen-sensitive cells, and these temperate phage out in the environment. And you'll notice there are some double use of T. These are these sort of delayed differential equations I described before. And you don't need to see, I mean, you can look at the details. In essence, the cells are taking up resources. The temperate phage are going inside, sometimes killing, sometimes becoming lysogens and popping out. And they had a premise, which is that temperate phage do better when few hosts are available and extracellular mortality rate are high. But in spite of the intuitive appeal, we're unable to find solutions to it. And the general dogma has been, to answer your question, what is the adaptive significance? So why bother if there are many viruses around that are co-infecting the same cell? The idea is, well, if many viruses co-infect the same cell, it's likely that there aren't that many other cells out there that aren't yet infected. So if you kill that one, and that's happening everywhere else, we'll kill off the cell population. But it appeals to some notion of group-level selection that somehow they're aware in some ways. So I'll go back to that, but that's been the dogma, but it hasn't necessarily been resolved. There's a neat story by Rotem Sorek on the Howe side of small peptides that seem to be released upon lysis that have changed and modify the cell fate when the next virus comes in, but that's something that's come out in the past year or so.
I guess it's an alternative. It's just that it's a consequence of having, as you said, shared regulatory system.
Yeah, so we've constructed, and again, I moved those slides away, but we have constructed a model in which these shared regulatory systems, you can get outcomes where more virus means less virulent outcomes. And now just in the way that you gave this talk, there's a sort of a hyperparameter, and then there's a selective event. You could imagine that you could evolve those gene regulatory networks, and that would be the adaptive thing. But certainly from a how point of view, you can make that happen. Does that satisfy the why question? I don't know. So I'll try to get there in this talk. Okay, so viruses kill. I've given you some basic biology of how they integrate and showed you that at least in these model systems like phage lambda, people have studied them, and we know a little bit about the ways in which the outcome or the switch can be modulated. And it can happen certainly in some cases, but again, my interest has been to try to understand the relevance of viruses in natural systems. So I want to know to what extent are these curiosities that we can learn something from an evolutionary sense, or are they really ubiquitous in the environment? And part of the challenge there is that a lot of the ways we look for viruses, we look for particles, we count particles, right? And we look for lytic events. So all these things, all these phage bacteria infection networks are looking for spot assays, killing. So most of the work is biased in some sense towards looking for the lytic option. In the past few years, there's an increasing recognition that latent infections, lysogeny, maybe in forms of chronic infections, are increasingly the norm in environmental microbiomes, meaning in marine systems, as well as in mammal-associated microbiomes. So I've given just a few papers here. If you're interested, you can see the titles. Lysogeny is prevalent, widely distributed. Here, this is a mirroring gut. This is from the North Pacific Ocean, which is, as you go down in depth, integrase, C1 repressor excisionase are all hallmark genes associated with lysogeny. So they're finding evidence inside microbial genomes of a lot of different prophage doing it. Now, they don't know what they're all doing, but that there's evidence that things like prophage are integrated inside the microbial genomes. And this is one that just is on the archive. I bring it out only to say that there's a major important class of cyanobacteria, which are these autotrophs that live in the surface oceans, that act in some ways as microscopic plants. And until this past year, no one had found a prophage of this variety, and now all of a sudden, now that people are beginning to think they're there, they begin to find them. This is of Synechococcus. So they're out in the environment. In fact, at least without knowing the details, people have known that lysogeny can occur in marine environments for quite some time. So there has been a dogma in the field going back into the 90s and a little bit into the 2000s, and I'll give you just two examples here. This is work in which Jennifer Brum and colleagues went and looked at Arctic Ocean environments in the spring and summer and looked inside bacteria and said, what fraction of them are lysogens at any given moment in time? And you see there is a very big difference here between the relatively non-productive or low productivity periods in which you see a lot of lysogens and the high productivity periods in which you don't see that many. And in contrast, when they look at the percent of infected cells, actively infected cells, so undergoing lysis, you see not that many, and all of a sudden the switch. And she called that a seasonal time bomb. That when times were good, when there was a lot of nutrients, a lot of cells in the environment, then they seemed to see more lysis. And when times weren't so good, then you seem to see more lysogeny. And this goes back, in some sense, to a similar argument as one might expect, even from the phage lambda story. And this had gone so far even before. This is Laura McDaniel and others at University of South Florida, who consider lysogeny as adaptation to poor host conditions. So occurrence of lysogeny is at times of low host availability, resource limitations, or somehow adverse environmental conditions. And they did this by looking at the relationship between, in essence, productivity as measured somehow by primary productivity. You can use chlorophyll as a proxy or so on, and finding that when it was high, you didn't find that many cells that could be induced, so you had low lysogen fraction. And when it was a low productivity environment, they found there was, relatively speaking, a high fraction of lysogens. Okay? So this was the dogma until about 2016, just a couple years ago. The idea that when there are many hosts around, you're going to see a lot of active infections and killing, and not that many hosts around, you're not going to see as much. You're going to see more lysogeny, which is really what the phage lambda story says too. And in 2016, a group proposed something called piggyback the winner. And the piggyback the winner idea is that, in contrast to everything I just told you, lysogeny is positively correlated with increases in host density and productivity. So the more dense or productive the environment is, the tendency should be for viruses to have a preferential lysogenization or increase in lysogeny, and relatively speaking, a decrease in lytic activity. Okay? And one of the supposed consequences of this model is that as one increases the number of microbes or finds that concentration of microbes go up, the number of viruses per microbe should go up, but not linearly. It should be sublinear. And realize that's a subtlety. These are log-log plots. So instead of it going up for a doubling of bacteria, instead of doubling the number of viruses, maybe you go up by 1.5, right, or 1.2. And if you double again, you don't double the number of viruses. You go up in a sublinear way. And the idea was that if this were happening, then this would happen. And I should point out that we've looked at similar data too. There's other groups who've looked at it. I know it's a busy slide. These are one-to-one lines for a whole bunch of different systems. There's this common line, but you'll notice that the lines tend to be shallower in slope. Each case, it's virus abundance, microbial abundance. So there seems to be a general trend across many systems that as the number of microbes increases, viruses increase, but less than proportionately. And I have to explain sort of why that even mattered. There was this dogma that there were 10-to-1 viruses per every host. But if as hosts increase and viruses don't go up as many, there's not one number. There's not a notion of a constant. In fact, the ratio of virus to microbes should go down as microbes go up. Okay. So this theory was in some sense stemming from the fact that there was this unexplained observation that seemed interesting, and also because at the same time we were seeing a lot more lysogeny or evidence of lysogeny in natural systems. And they managed to connect these two ideas. We started to look into this a little bit more, and we became a little bit concerned, but also interested. So I'm going to kind of tell you a little bit here about both our concern and our interest. This is their model of the piggyback the winner system. You'll notice it has cells and viruses, but no lysogens, which concerned us, because it's hard to have a model of lysogeny without infected cells. You have microbial growth, lysis, and that P should be a V, excuse me, mortality, the viral particles decay, and they had a notion here that instead of the viral release just being a constant burst size, that it would be modulated by N over K. N over K would seem to say that as N increases, there's more lytic activity. So it's kind of exactly the opposite of what they had to say. On the other hand, it produced the right pattern. And if you want to read more about this, it turns out many models without lysogeny can produce this decreasing pattern. And so we felt that this pattern was not a hallmark of this piggyback the winner, but rather one should really look more carefully at the underlying evidence of the association between indicators of lysogeny and microbial abundance. So we began to do that. Here you see the evidence from the paper of microbial cell density on the x-axis and pro-virus-like reads in the virome, and these are all from coral reef studies. This is the original data that they had said was positively related, that more microbial cells meant more evidence of lysogeny. We don't think so, right? So First of all, it's a non-significant correlation, and to the extent to which you remove a few points, even if you were to do that, the explanatory power is very, very small. That's provirus-like reads. These are integrases, same story, and this is excisionase. So we concluded instead that there was an absence of evidence. There was certainly lysogeny out in these systems, and evidence or proxies of lysogeny, but there wasn't necessarily a clear signal. And also there wasn't a clear signal the other way, and I'm happy to talk about that as well, right? I should point out here, they're looking in the virome, so they're looking outside cells for indication that these could be temperate viruses. And there seems to be an absence of evidence of any correlation between the microbial cell density and these indicators. Okay. And this remains something that, as you can imagine, when you have these back and forth responses, it remains of interest to a number of folks. And in fact, this past year, there was a follow-up where people began to test this idea of the piggyback the winner. Here's microbial abundances. Here's the virus-to-host ratio. And as I told you before, one of their claims is that the piggyback the winner should lead to these decreases in virus-to-host ratio with increasing microbial abundances. Right? I think I've told you that. There's a lot of ideas today, but I just said that one. Right? So we increase microbial abundances, and we expect a decrease in virus-to-host ratio. And they, in an independent study, found many, many relationships. Each line has a different virus-host pair at the genus level that they've putatively assigned from metagenomic data. So they went into the environment, identified virus-host pairs, and said, these are the pairs in the different samples. What are your abundances, and what are the patterns? So this seems quite good. I don't know if does anyone see something interesting here? I don't know if I've given you enough. So the x-axis is the microbial abundances, and the y-axis is the ratio to virus to microbe abundances. And the prediction is supposed to go down. Anyone see? This is sort of what we're supposed to do as scientists. Look at stuff and think. So I'm going to try to just make sure it's clear. The y-axis is the ratio of the viruses to hosts. Is it helping at all?
It should be like less than 1. I mean, just from n, the dependence on 1 over n over there, you expect that minus 1 is the...
Right, so when I looked at them, because I've been following this and trying to see what other evidence, because maybe our response was wrong, and in fact, this piggyback the winner made this relationship, and I need to be thoughtful about how these systems work. And so the one thing when I see such plots is that there could be a negative relationship, and this would be an appropriate way to plot things, and it's hard to see the axes, but you can see that the thing you don't want this to be is a -1 slope, because when you take a ratio and plot it against itself, you will find on a log-log plot, it looks like a slope of -1. Whereas if there was a relationship between viruses and microbes but was sublinear, you'd expect these to be shallower than -1, right? So I'm going to show you now a -1 plot, and you'll notice that it is quite parallel to a lot of these lines. So we began to reinvestigate things and just ask the other question, which is, are virus abundances related to microbial abundances in any way? And the answer is no, they're unrelated for almost all of these. So if you want to hear more about another debate that I've managed to pull myself into, you can see this in about a week or so. Okay, so that's some of the background here. But both the original piggyback the winner and this model and some of their analyses do raise important points of observations of latency in the environment, but I think they raise another important point, which is, I don't think we really know the answer to this question. What environmental conditions should favor lysogeny rather than lysis? Okay? I understand the piggyback the winner has one idea. Some of the work on phage lammas suggests the exact opposite, or the seasonal time bomb suggests the opposite. So I think that's an opportunity to take that confusion and try to ask some systematic questions. So as a theory group and a modeling group, and having thought about this for a while, I want to try to at least provide a framework to begin to, what I think is a productive way to answer this question, or at least begin to make predictions that we can then go test. And to do that, I want to begin with this old idea, which some of you may know. It's an adage, at least in English, a bird in the hand is worth two in the bush. I don't know how to say that in Portuguese. Someone can say it. Okay, in the sky. Okay, because people are doing something. Flying, okay. Right, so the key idea here is that if one has something right now, right, how many are you willing to trade it for out that, some benefit, but you notice it's two, it should be more than what you have right now because there's some uncertainty. So I want to take that idea and suggest that the puzzle that I'd like to solve is the following, right? A virus in the cell is worth N in the bloom. You're a virus. You have the cell. You're an obligate intracellular parasite. You got to find one. How many of you out there is it worth to abandon this cell? So in essence, what is N? Right?
Let me answer that. In Colombia, it is 100 in this category.
Okay, fine, 100, right. So you can, this may be an interesting cross-comparison culturally of how much instability is in different countries based on what that adage is. So that may itself be, that could actually be quite a good essay. You should go write it. Okay, so, right, what is N? And I want to now bring it back to the notion I had from the very first series of lectures and connect it to latency. So here I've provided a sketch of the environment in which we have these susceptible cells, a virus, and it injects its genetic material into the host. And I'm going to call this the mother virus for the moment. I haven't used that word before, but I would like to use it now. Right? And it seems strange to talk about mother viruses and baby viruses and daughter viruses and so on, but I'm going to use this word mother virus. After a certain amount of time, which I explored at depth in my first two lectures, there will be a lysis event, and many viral progeny may get out, let's say 100. But if that's the mother virus, those viruses, viral particles, I won't consider that fitness. I won't consider that the viral fitness of that mother virus, because it could be that 97 of those virus particles never find a host cell. Tree and seeds. Many seeds go out, but only a few of them are going to establish. Here I have an example where 3 of them were lucky and managed to have essentially the progeny of this mother virus, who then themselves become new mother viruses. Yes, they're the daughter of that mother virus, but they themselves could have progeny as well. And in that sense, from the fitness perspective of the average number of offspring, or the number of offspring of this particular individual, I would say the answer is 3. That the individual level fitness of that mother virus was 3, because it created 3 new mother viruses in its lifespan before it died. I'm using a subscript HOR for horizontal, because in my view, this fitness was derived from a virus finding a new cell somewhere else rather than sticking with the current cell. And now I want to contrast it and begin to maybe make some headway on the puzzle that I had. I don't know if it's a puzzle for you, but certainly this I struggled with, which is, why would it be worthwhile for the virus to stay with the cell? Because as before, I had been counting fitness over here. You start with 1, you make 100, and when you make an integration event, there are no viral production around. There's no production of new virus particles. So here's the same setup. Here's entry, here's the mother virus. Yes, it could lyse and do exactly what I described before, because these tempered phage have both options. But in this case, the mother virus divides, made a daughter virus, which herself becomes a mother and has her own fitness, and then divides a second time, divides a third time, and eventually dies for whatever reason. So I claim that now, even though no virus particles have been made, that the fitness of this virus is 3, because 3 new mother viruses were generated by this mother virus before it died, in other words, in its lifespan. And so I say 3, but I'm put VER to denote vertical. The idea that the fitness was derived by sticking with the cell and vertically passing on material to daughter cells. Okay, so here we have 2 vastly different strategies. killing your cell, finding a cell somewhere else, and sticking with your cell and producing daughter viruses, which then have their own fitness, can lead to the same individual level fitness, right? So how does this depend on context? How does this depend on cell densities and on the environmental conditions? Yes?
So when there's the vertical transmission, that's when the host cell divides itself.
Correct.
How can you keep track of the mother, like the mother zero? If like there's a cell division, which one is the mother?
So I do know what you mean. So we'll go back. There is some work on cell aging, right? But I concede the fact that I have to choose one of the two, right? And so in the absence of information, I'll make that arbitrary choice. It'll still apply. I can't choose both. I have to decide this is the mother. Now, in fact, the reality is that there is some notion of aging. Lin Chao has worked on some of this, but I'm not predicating here. As long as I make a choice that only one of them is the mother, one of them is the daughter, then the logic applies. I can't have them both be the mother because then the daughter fitness has to be its own. And it could have its own fate. It could then have an induction event and create 100, then it would have horizontal fitness instead of vertical fitness. It's a good point. Okay, so how does this depend on cell densities? Right. So what I want to do now is connect this individual perspective with the population perspective. And I'm going to do this by connecting the same mathematical ideas I did earlier in the week, but then extending it into these new domains. So again, this is what I just described. The horizontal fitness of this particular individual is 3. Here's a model in which we have susceptible cells, infected cells, viruses, and these viruses are going to be obligately lytic. They can only kill the cell. And as before, I have logistic growth, infection, we have lysis over some time, and then producing beta new viruses. And so the question at the population scale is, will, when I add a virus, it increase in abundance? Will it invade? And as I already showed you earlier in the week, viruses increase in population at the population scale, given exclusively horizontal transmission when this value is greater than 1. And again, let me walk through the pieces if you don't recall it from before. I'm going to start, and I try to explain this in 2 ways. I want to start from the perspective of the particle, imagining that that is the right way to calculate fitness. So if I start from the particle, I need to complete a life cycle. Right? So if I have mother particles, then they need to go particle, cell, back to particle, and I should count. So if I'm a particle, the probability I find a cell before I decay is the adsorption rate divided by the absorption rate plus the decay rate. Because that's the probability that that virus particle adsorbs before it decays. Now, once inside, this is the probability that it lyses the cell before the cell dies or is removed for some other reason. So get in, get out, and if you get out, produce beta things. That is a dimensionless number which says, on average, how many new particles does one particle generate when introduced into an otherwise susceptible environment. But I can also turn this around, and actually this number had been introduced and is called the phage reproduction number in the literature. And I can change my perspective and think again about mother cells and start with an infected cell and make the cycle be cell, particle, cell. Because if I use a cell-based metric, I can start to compare apples and apples to my other strategies. And in that case, if I start in the cell, this is the probability it gets out, makes this beta of the progeny, this many survive, and we have the same answer. Just a different interpretation. Okay? But you'll notice here that the invasion fitness, or this horizontal basic reproduction number, depends on the availability of hosts. I just said all that, yeah. Burst size, so I think I've explained it. Any questions about this? So here we have burst size from 5 to 400, and this is the susceptible population. And this is log 10 of that horizontal R naught. So for a given burst size, there are regimes in which there are not enough cells for a single virus on average to invade, even though you're producing 100 progeny. Because of those 100, too many are decaying before they adsorb. But yet, there's a critical point, which if you notice, this is the line that divides them. And as the intracellular period is more productive, you need less cells outside to invade. And as the intracellular period becomes less productive, you need more cells outside. So this suggests that as cell density increases, right, it's going to favor, all things else being equal, invasion. Okay? If you go this horizontal route. Okay. Now let me do the same thing with the latent route. This is the model that I described before. Here, there can be a latent state which needs to induce and produce new viruses. And in this model, I'll call these lysogens. We have infection. There's lysogen growth. I have both lysogeny and lysis here. And when there's a lysogeny component, then lysogens can grow. And I'll just make one note here. B prime is the birth rate. I have a value Q and P. This just allows me to sort of play with the probability that you're going to kill the cell versus stick with the cell. And I'm, for the moment, going to ask, what happens if it's exclusively vertical? It turns out the mixed case in its full glory is a little bit more complicated. So Q is 1, P is 0. There's not going to be any initial lysis. So if one virus shows up in a susceptible host population, will it spread? It's going to spread whether or not the lysogen can make enough daughter cells. And how do we estimate that rate? The vertical fitness, which here is 3 in this example, is this expression, and it comes right out of analysis of these models. What does this mean? B prime 1 minus S star over K over D prime. D prime is the cell death rate. So what is 1 over D prime? The average duration of infected cells, the average lifespan of infected cells. What is B prime 1 minus S star over K? That's the growth rate, or division rate, excuse me, let me say division rate of infected cells. So you have a division rate times a period over which you're dividing. That gives you the average number of daughter cells. And that's what I just said. So if the average number of daughter cells is greater than 1, you never need to make the particles for the number of infected cells to increase. But again, this depends on context. Here's susceptible host population. Here's this basic reproduction number. This is the one I showed you before for the horizontal case. Right? So as you have more susceptible cells, you're going to have an increasing horizontal fitness. This 3 curves are 3 different possibilities where I've modulated the value of B prime. Do lysogens grow faster or slow, or a little bit faster or slower than do uninfected cells? And when there are some fitness benefits of lysogeny, in other words, that infected cells grow faster, then it turns out that at low susceptible cell populations, this vertical pathway is beneficial. This, by the way, is the number 1 here, this dashed line. So if things are above 1, they're going to invade. Why would it be better for a latent infection, for there to be fewer host cells around? If one infects vertically, then your fitness depends on reproducing as a cell with a prophage inside. The fewer cells around, the less competition you feel from the susceptible cells. So for example, if there are very few susceptible cells around, you're a virus. If you knew that, and there were sufficient resources, which I'll get into later, then do you want to kill that cell, and there's no cells to infect, or do you want to proliferate along with the lineage of the cells? And it turns out that in terms of, if I count the number of virus genomes inside cells and call that fitness, then fewer susceptible cells is actually better for the latent pathway. And that's true even when there are low benefits of lysogeny, even when there are fitness costs, but you have to have still a critical number, depending on the fitness cost, for there to be invasion. Any questions about this part? Yeah.
N is L plus S.
Right, N is L plus S, yes. So they're both feeling effects, and we're assuming it's equal. That could be modified too. Correct, thank you. I should have said that. Okay. So now let me go to a third option, which I haven't talked about, which are chronic infections or chronic viruses. So I've given this option of killing, and the only way to get to the next cell is to kill. But as you probably know, in other examples, viruses can bud out, releasing progeny without necessarily killing their host cell. And in that way, a virus potentially can get the benefits of both pathways. It could reproduce along with the cell, or concurrently also release new virus particles which can go out and infect other cells. So I'm going to use a similar model where we have susceptible cells, infected cells, but now they're chronically infected, and viruses. And again, we have this entry point. Let me tell you the individual perspective, which I haven't explained yet. It's this mother virus. This mother virus divides, so now we have one progeny. And at the same time, it has budded off viruses which horizontally have found new cells. Not all of them make it. So it divided once. It had 2 of its progeny viruses find new cells. So now its fitness is 3, just like it was before, but it's decomposed into vertical and horizontal components. Yep?
What's the difference between this and the previous? Is it just that you have the decay is slower?
Yeah.
You're dying the particles. So what does the encapsulation do for them, other than just allowing them a longer time in the environment?
Okay, so let me just clarify. The first model was the virus particles in all of these models are the same. So they're particles, they go out, they find new cells. The means by which you release particles are different in this model. And maybe you'll see it here in the equations. In the previous model, in order to release particles, even you have to kill it. Here, although the cell dies, you notice this virion production can just happen. Right. It turns out that you can make a remapping of these two models, but when we put in the full details of latency, then they actually diverge. But the key thing is that release of virus particles in this model does not require death of cell. And so here we have logistic growth of the two, infection, which leads to these chronically infected cells. Cells can die, viruses can decay, but differently than before, there's not a burst event, but rather there's a background virus production, right? Okay. And so in this model, we can calculate, just as before, the basic reproduction number for these chronic strategies. And it includes a horizontal component and a vertical component. When this is greater than 1, we expect then that the infected subsystem components here, right, because there are viral genomes in both these sides, will increase in abundance. And how do we understand this? This I've explained before. It's the average number of daughter cells produced before that chronically infected cell is removed, dies, decayed from the system. What is this horizontal? Let's start from the perspective of inside the cell. The cell is going to last, on average, 1 over d prime. So in that period of time, how many new infected cells will it generate horizontally? Well, it's going to generate alpha per unit time. Of those alpha, this many survive. Multiply by the time, and you get the average number of horizontal transmission events. Okay? Now, it turns out, seeing these after the fact and calculating them in advance, once you get used to these systems, and part of what we've been trying to do is make it simpler to make these kind of calculations. These are not just me hypothesizing. These are what come out of a linear stability analysis of these systems, or, as I'll mention, if you're interested in doing these yourselves, called the next generation matrix method. So there's a way to do these calculations in a formal sense, but there are some pitfalls. Okay, so now I'm going to combine the 3 ideas together. We have susceptible cell populations that are increasing. We have the basic reproduction number, whether it's vertical, horizontal, or chronic. And you'll see that the vertical goes down as we have more cells, because when there are fewer cells that are all susceptible, then you can spread along with them and without as much competition from current cells. When there are many cells around that are susceptible, then the horizontal strategy is best. In other words, the obligately lytic strategy is best. And here we have a trade-off so that the chronic strategy is neither good in the low or high, but can actually exceed both in an intermediate regime. Right? So the point here is that the context, ecological context, can be strongly informative with respect to which strategy is more adaptive in the sense of invasion fitness, directly coming out of thinking about viral fitness in terms of infected cells and not particles. And I want to point out that there's some old work that we're trying to engage with here from Thomas Berngruber and Sylvain Gandon, and it's not that old. It's 2013, not 2003. I missed the number 1. And here they did some experiments with phage lambda and E. coli, some which were obligately virulent and some which could enter lysogeny, in other words, the wild-type temperate phage lambda. And you'll see here these are proviruses, so viruses that they see inside cells, and here are free viruses. The red is that they started the experiment with very few infected cells, and you'll see that in those situations, the ratio of virulent to non-virulent kinds was high. In other words, when there were very few already infected cells, a lot of susceptible cells around, it was better to kill. But when they inoculated the system or started the system with many infected cells, then it was better not to kill. Right? There was no benefit of going and killing the cell and finding new cells because they were already infected. It turns out there's superinfection immunity. And that applies to both cases. A lot of noise in the system and there's other things going on. But just to suggest that at least some of these experiments have been done, and I think it's time to go back and do more. Okay, so at the middle portion of this talk, after giving some of the context and the reason why I'm interested, I asked this question: what environmental conditions should favor lysogeny rather than lysis? I tried to ask this question of, a virus in the cell's worth N in the bloom, and what is N? And I think the answer to what is N is there's not just one number. It actually depends on context. But throughout this talk, I've used this definition, and I just want to define it so it's clear, that I think the appropriate way to consider viral invasions across qualitatively different strategy types is to think of the average number of new infected cells produced by a single typical infected cell and its progeny virions in an otherwise susceptible population. So in this approach, I'm adopting a cell-centric fitness metric. And using this, I can think of a strictly lytic type, because then it's a newly infected cell, but you get the benefit not just of that cell, because that cell doesn't produce a newly infected cell, it's killed, but rather all the progeny virions are included in the calculation of that particular fitness. Right? Okay, and just if you want to see all of these together, if this is something you're interested in, I just want to put these compilation of the 3 contrasting examples. And this is, if you're interested in seeing more on the bioRxiv, the paper's under review, but you can see all of these same results and the details of how we did the calculations. And I just want to make one side comment about the technical note. If I'd had more time, I could actually talk about some of these issues, but it takes a little bit more time to get through. There's an idea of the next generation matrix method popularized by Diekman for applying to epidemic outbreaks. If you want to look at that, it's Royal Society Interface 2010. It turns out these equations are very readily interpretable. They don't get so interpretable, and one of the things we think we figured out is how to make them interpretable even when you have mixed pathways. So right now I've had strictly lytic, strictly latent, and this sort of additive combination. But it turns out that the real phage lambda circuit involves the possibility of integrating and coming back out and then reaching the cell, which is a combination of vertical and horizontal pathway at the same time. And then these calculations don't look so nice, but you'll have to check in about a month. We're wrapping that up, and we'll put that on the bioRxiv, and hopefully that will help too. So let me go back to a little bit of a, yes?
So you don't consider the possibility that a latent virus enter into an infected cell and an interaction between the two mechanisms?
Yeah, so we have right now in these, there's 2 things to keep in mind. There's a difference between short-term and long-term fitness, or short-term evolution and long-term evolution. In this case, we're looking at the outset of the infection, does one invade? So a lot of what we're doing now, you have to check in with me later, is to ask the question of what happens once the virus population has already established itself. But even in that case, the question of multiple infections is relevant.
Because
Yeah, so we've seen, I'll show you something, a first step towards that in about 2 slides, but it's not complete. But you're right that if you don't have superinfection immunity, you can imagine these lysogens are doing very well. In comes a new lytic type and can just take over the cell, right? Maybe with consequences for later. I'll show you one example. We don't have a complete solution yet. Okay, so where's the, right. Yeah. Right, so the way that when we do these calculations, you have to think of epidemiological births, so there's transmissions and transition events. And so we view transmission events as being the introduction of a viral genome into a cell. And transitions could be from an eclipse phase to an infected phase or the integration. So those are all transitions that don't lead to new fitness. So we have to differentiate between transmissions and transitions. It gets a little technical, but you're right. So that, and even the production of virus particles, we don't think as a birth or it's a transition in the status of the virus. Maybe that wasn't your question.
When you calculate the reproduction numbers, you have to separate into matrix, right? When you calculate by...
You're actually talking about, right, so I was trying to give you the answer. So we have the transition matrix and the transmission matrix. The transmission matrix for these systems, where we call an epidemiological birth, is only one state, which is the infected cell. Now, when we do the full model, which I was about to show, there are 2 ways, and that gets more complicated. It sounds like you know something about this method. That's another, but you're right, yeah. Okay, so, right. We're getting a little bit late on time, but I'm nearly done. If and I think maybe even last week when you were at the course, if you recall, you probably learned about epidemiological dynamics and were told something about R naught, which is supposed to be an epidemiological concept. And I started these whole lectures by telling you viruses were predators and their hosts were prey. But remember, they're obligate intracellular parasites. And so some of what I'm trying to do today and what I think we're going to be trying to do in my group in the next few years is unify these ecoevolutionary studies with epidemiological studies. So really try to combine these together. And just to point out again, did you all go through this? It's the same concept. And so, okay, if you've seen this all before, then I don't need to go through it. Then I'll just skip through it only to say the following, that if you look at the invasion of a infectious disease in a population, we get exactly these same concepts, that the increase in infected individuals, in this case humans, grows at some rate, but that the condition has to do with the transmission rate times the average infectious period. It's the same idea. How many new infections does a single infectious individual typically create in an otherwise susceptible population? Right, which is what I just said. And I want to say one more thing about that, which I think is relevant, and I think my computer thinks there's no more. It says your system has run out of memory. It's too bad. These are 3 examples of outbreaks with the same value of R naught. You'll notice they look exactly the same. And I just want to make the point, these are 3 dynamical simulations, that there's a difference between population dynamics and the criteria for invasion. And it's possible with the same value of R naught initially to have very different long-term outcomes. This is also why we think we need to start looking at long-term outcomes, not just the short-term. And for those of you who are not familiar with these ideas, this is a perspective paper which explains the reason for this apparent paradox, that things with different strengths all have the same speed initially, but they have different final sizes. Okay. So let me go back to what Stewart and Levin asked. They had this premise that temperate phage do better when few hosts are available and extracellular mortality is high. Although that didn't come out because they were looking at these numerical models that are a bit more complicated, this directly comes out from thinking about fitness of infected cells, measuring with the cell centric idea, because then you can see when few hosts are available, even if you kill the cell and make a lot of viral new virions, there are no other cells, or not enough to infect. And likewise, when extracellular mortality rate is high, that's going to mean you're more likely to decay than to reach that new susceptible cell. So it exactly falls out of these models. And so what we've done, without going into the details, we've taken their more complicated model of a resource supply, uptake, infection of this lysogen, which can be induced, and then look to see how does R naught change as a function of both the population and the resources, and asking what would be the optimal value of P, the probability of lysogeny when you get in, and gamma, the rate of induction. And it turns out these blue types are these strictly latent types, and these are the strictly virulent types. And what you see is that given a constant level of resources, the more cells that are available, it's better increasingly to become lytic. But interestingly, now there's a transition point I'm just showing up, that for a fixed level of cells, as there are more resources available, it turns out to be better to not kill. Because as you increase the amount of resources, this is a resource-explicit model, now if you don't kill it, that cell may grow faster than you would have otherwise gotten the benefit from going horizontally. And there's some other neat things here that show that the best thing to do is to be strictly one or the other. It turns out that that's probably not right because, first of all, the virus doesn't know exactly what these values are, and also the values themselves change because a strictly lytic population of viruses are going to drive down susceptible cell populations, turning it into an environment that potentially it may be better for lysogens. And that's something that we're doing too, and the reverse. So to answer, I think, your question, Matteo, is that we tried to answer this in a different way. Could lytic viruses help chronic and temperate viruses by reducing niche competition? It turns out you can play the game in both ways. So we have these susceptible cells, and we have chronic infected viruses, which we've introduced, but are going down. It turns out that without these lytic viruses, they will disappear. We introduce lytic viruses in this model. The lytic viruses reduce susceptible densities, and therefore release some of the niche competition from these chronic cells which recover. So we can go from a situation which a chronic cell on its own wouldn't make it, but if a killer virus is there, it actually helps the chronic virus because it reduces the competition. We can make things work in the other way too. And I should point out that there's been quite a lot of interest in this issue of different strategies at the same time, and this is very speculative. These are viruses detected in genomes with signals. These tend to be double-stranded DNA that are thought to be lytic, but the strongest co-infection signal, and I should point out there's a lot of co-infections, tend to be from these filamentous single-stranded DNA viruses that are thought to be chronic. So we're beginning to see evidence, at least in natural systems, of ubiquitous co-infections, including different modalities, and we're still scratching the surface. But this is beginning to answer your question about what happens when two strategies intersect. Okay, so that's more or less it. Just to give some perspectives, that I think the evolution of lysogeny and chronic non-lytic infections remains understudied. Both in the environment and even from just these fundamental perspectives of what happens in simple environments. Here I've tried to explore ways to examine fitness of viruses in terms of an infected cell-centric metric, thinking about how many cells are infected rather than how many virus particles are produced. Virus particles may be a route to have infected cells, but they're not an end in and unto themselves. And keep in mind, of course, that these are obligate parasites. The unit of selection is the genome, not the particle per se. This framework, I think, can help reveal why lysogeny can outperform lysis, at least in the short term, without appealing to any other ideas of maybe in the future times will be bad. The other major point I want to say here is, right now, at this moment, it may be better for a virus not to kill because of the current ecological conditions as opposed to a future ecological condition. And we're trying to answer a lot of these evolutionary questions in the long term with co-infection and so on. And I'm happy to chat more. I mean, I think this is really just the beginning of what I hope we can do moving forward. And then there's a last question that I've been struggling with, which is, again, in my mind, when I think of virus, I think of this. And there are a number of people who had Patrick Fortin in particular has had the notion that there's something called the virocell. So an actively infected cell, which is making virus particles, is the real mature virus, and this is the seed. But yet when we do our calculations, this, which doesn't look very active in terms of doing much for the virus, is certainly in our perspective also counting towards viral fitness. And so I don't think one of these is the answer. I'm increasingly thinking of viruses as having a life cycle. When I need to think about fitness, I need to think across the whole life cycle. And it turns out that the life cycle may be involved going here to there, or it may just be looping there for a while. So these are really the kind of questions I think for the first time in quite some time that I and others may be able to make some headway on. And with that, we're supposed to take a break, but I think in lieu of the fact that people would like to go to lunch and let's just have continuity, we don't need a break right before lunch, I'll just take questions now and we can break in the next 15 minutes or so. Great, thank you.
More questions? We got the microphone here.
So you started by discussing the decision of viruses, whether to go one pathway or the other, but then in the model you discussed, there is no decision.
Yeah.
And also there is no communication between viruses. So are you saying that there is no need for either, say, viruses to take decisions, or otherwise, would there be some experiments that would distinguish just a purely population dynamic effects, like the one you described, from a situation where, say, a virus at a certain point decides whether to turn lytic or lysogen?
Yeah, so I think the brief answer to your question is that I think our work is incomplete. And I mean that in a serious way, which is that when I first got into the problem of lysogeny, I viewed it through this how question. How could viruses end up, through some intracellular process, lead to having their outcome depend on the multiplicity of infection? And I have some backup slides which explains it, but I don't think I need to go there. But the current model, right, so I view that as a next stage. So I just wanted to, even before going there, because in some ways I knew a little bit too much about the how, we could build a model in which there's really a gene regulatory feedback, there's a hyperparameter of that, and could you evolve those hyperparameters that lead to some decision that's adaptive, so that in some cases, when there's multiplicity of infection, then you see lysogeny being preferred, and in another case not. And I think that is a nice way to go. But the point of this is that I first wanted to even establish how do I measure fitness and why would, at the moment, not killing your cell be better. So we've started here. But I did keep that in there, not just for historical reasons, but because the central focus of a lot of the work on latency is this how question. How do viruses both sense the cell and get out? It's the basis of the genetic switch. There's a book called Phage Lambda Genetic Switch by Mark Potasio. So it's really the centerpiece. To have the right evolutionary framework is necessary, I think, before we start embedding those details. So that at the moment, it's incomplete. We haven't done it yet.
Am I right to say that the work that you have presented would be as if this decision is written on the DNA of the virus at the moment, and you have two different populations that inherit this trait?
Yeah, so is it written on the DNA of the virus? So at the moment, we have a P, a probability of having one thing or another, or a gamma, which are fixed traits. And you can imagine the offspring having a different fixed trait. The reality is that the cell status could make a difference. There could be other small molecules. So you can imagine even that could be some sort of plastic trait, which gets some notion of cell status. The work of Rotem Sorik is one indication. But to do that, then we'd have to extend what we've done and not just have it to be a fixed trait, responding to cell state. And you could imagine in some ways what we're trying, and that's where we're trying to go, is that I think that the P is not a fixed number that in reality that each phage contains, but rather It's a gene regulatory circuit. Maybe I keep talking about it. Maybe I need to say something about it. Right. So in the bottom left is a little bit of a small pictorial of what one of these decision circuits are. They're essentially a bidirectional promoter, a regulatory mechanism inside phage, in which one pathway says lysis and one pathway says lysogeny, and essentially there's messenger RNA which gets then translated to protein. They combine back at these operators, and there's a positive feedback loop that reinforces decisions, but it can then exclude, so it's bidirectional. There's a stochastic element to it as well. So you can imagine that if this is the actual decision circuit, this is really what those phage are carrying. And you can imagine a simplified version of this that becomes really the thing that evolves, not the value P. And just like in I think, Paul Rainey's talk, the selection is delayed till later. It's not being selected on these, it's being selected on whether or not it can create new viruses. So the P is the feature that's going to dictate whether or not you get to the next cell or to a new cell horizontally. But it's arising through these mechanisms. For the moment, we haven't created an abstraction of that we've embedded in the model. Does that help? Yeah. I think in the back there's one.
So, I mean, you briefly commented on something that's coming up where you take into account some transitions, right? From lysogeny to lytic strategy. Can you comment a little bit on that? And maybe maybe a better question would be, how would you extend that R naught metric? Like in your mind, what did you see would be a good extension in order to capture these long-term dynamics?
Yes, so there's two parts there. Let me try to answer the first part, which is we're wrapping up something in which we really are thinking about two ways to have epidemiological births, and I think that's what your question is there. So here I've just had one way, infected cells. So when you think about lysogeny in the complete life cycle, there can be two ways to have a birth. One is that the virus comes in, and I should probably just draw this. The virus comes in, and let's call this an exposed state. And then there's a decision, lysogeny or active infection. The active infection makes viruses, right? And we can think of this P as being something that results from a more complicated dynamical system on its own. This is driving new infections. So an epidemiological birth could be the introduction here, because E goes to I, goes to V, back to E, right? So that's one way. But once I have a lysogen, I don't need to do this anymore. I can have a birth here. And because they're disjoint, I don't need to go through those steps to do this process. It turns out that in the event that this happens, there's an induction event. Then there are different kinds of loops. You could have a lytic loop, which is E goes to I, goes to V, goes to E, right? And you could have a latent loop, which is L goes to L, just make it around. But you could also have a lysolytic loop, which is E goes to L, goes to I, goes to V, goes to E. And you could see why I didn't include that in my seminar today. And it turns out we can calculate the expected reproduction number in each of these cases, but you can't add them up. You can't add them up like the way I said, because when you do the next generation matrix method, it turns out because of these latent loops, you actually have to think not just of one generation, but it turns out 2 generations, and you have to discount in funny ways, and we think we've solved those problems, but it gets a little more complicated. So just to say that that's the answer, all my answers are too long. The answer to your first question is that if we really incorporate this, we have to think about latent loops, lytic loops, and lysolytic loops, which I didn't have to think about when I ignored the induction. Okay? And when we do that, it gets more complicated. And the second part of your question is long-term evolution. I don't think any of us have a good answer yet, but clearly there's an interesting short-term and long-term, and the most important thing is because there's feedback. If you have a viral strategy, then if infected cells are proliferating that are latent, and the latent cell way of regulation is not listened to by the lytic strategy, those are all susceptible cells. In your sense of Matteo, it's a defector. It comes in and just obliterates it. But once it starts to eliminate cells, it reduces its own fitness. And now with less viruses, less cells around, it may be, if there's still resources, an opportunity for latent types to arise. So we see opportunities there we have to do in the long term. Yep.
You present a new graph, a new analysis about the R, and I didn't understand well. And I didn't understand well. Could you explain?
Yes. Sorry, the computer really thinks your system has run out of memory again. This one you want me to look at? Is this one?
No, I think it's a curve about just a U. I don't know. You said that yes.
This one?
Yes. We saw this first three in last week, but this other one is like you said there is a paradox like that.
Yes. Let me just maybe I'll explain this. It's worth mentioning. There's a difference between the Malthusian growth rate and this R naught, which is the average number of new infected cells produced by a single infectious cell in an otherwise susceptible population. We call R naught often in epidemiological literature the strength, right? And often if you want to control an infectious disease, you're worried about a big R naught rather than a small R naught. R naught is very hard to measure. We don't measure it directly when we are doing outbreak control or even in these populations. It's the unknown thing that we hope our models can tell us about. What we measure instead are time series, and time series have speeds, right? So the point that I'm trying to make here is that here's infectious individuals. This is synthetic data in which I run a very simple SIR model, but it has a lot of analogies to what I've just done, with 3 different strengths. However, the strengths are associated with diseases with different durations so that the speeds initially look exactly the same. So there's a decoupling of strength and speed, and the last thing you have to think about is size. So these peaks here are all the size of the epidemic outbreak. And you see that the stronger disease has a bigger size and lasts for longer, even though it all has the same speed. And there are a lot of consequences of that kind of thing, but it really highlights 2 things. That first of all, you should know that there's that decoupling. But the other thing is that the long-term dynamics are not dictated just by this R naught. That dynamics matter and feedbacks matter. So I've been emphasizing invasion fitness, which is near term. This is to say that the long-term consequences are going to differ, right? And so we can't conflate the short-term evolution. Just immediately we'll say what's going to be the best over the long term.
Thanks.
Yep.
So with the evolutionary analysis that you did, can you have guessed or have you thought about which of the 2 strategies came first, like being latent or lytic?
Which of the 2 strategies? Are we in like a chicken and egg situation here? Which of the 2 strategies came first? If Eugene Koonin was here, he would have a good answer for that question. Yeah, I don't have a good answer. So, the short answer I think I'll just say is that I don't have a good answer to that question. I think it's a good question. And I was going to go back to these other things as well. I haven't looked at the evolution of the phage lambda switch. It's okay. It's on some random place. So I'd have to think more. I don't know. In the sense that it's interesting to think where the original viruses are always temperate and they lose that ability. If you go to symbiont literature, like do they get reduced in complexity when they start hanging out? I mean, it's certainly true that a virus that hangs out with its host cell, let me answer another question that I'll try. A virus that hangs out with its host cell too long, then there can be genome reduction and even loss of the ability to be an independent replicator. So that's actually something that I know Paul has another thing he has to do right now. But at the moment when the virus has infected the cell and they're replicating, their fates are entangled. But as long as it can still make horizontally generate virus and kill the cell, they haven't entirely been intertwined. Once they become intertwined, if it no longer kills the cell, then all of the parts of the genome that don't involve horizontal transmission are no longer selection. It'll get relaxed. They will disappear, and then you might just get relic elements. So with respect to at least some of the genome evolution, it's clear that viruses that hang on their cells for too long, you see a lot of these components that are now relic prophage rather than viable prophage anymore. That does tell you something about one sense of direction, but whether or not the original virus has always had this capability, I don't know. Yeah.
Any other questions?
All right. Well, thank you, everyone. I have one more lecture tomorrow morning. It'll be on going from theory to therapy. It's after Luca's talk tomorrow morning.
Okay,
okay, thanks.
Okay, so we come back here. We come back at 2:30. There are places to eat.
Yeah, so there's at least one place that we usually go, which is just at the end of the street where you go, well, it's Marquis de São Vicente.